So thanks for the opportunity to present some of our ongoing work that is trying to understand the mechanisms of cardiac microvascular dysfunction, specifically in diabetic induced heart failure with preserved ejection fraction. So diabetes itself really dramatically elevates the risk of HFpEF, and the mechanisms of HFpEF are still not fully understood, but it involves the stiffening of the left ventricle and defects in relaxation during diastole. But one of the features that has been noted more recently is that there is microvascular dysfunction in the left ventricle. This includes microvascular rarefaction, so less density of the microvasculature, as well as functional defects. And this is thought to be induced by a number of factors, including inflammation and endothelial dysfunction. And one of the triggers may be reactive oxygen species, which drives a decrease in nitric oxide, which normally would function to decrease stiffness and hypertrophy in the myocytes. This is also accompanied by fibroblast activation that really drives this fibrotic response in the heart. So we in the lab have been asking a couple of related questions. One is, what are the mechanisms that might be driving this microvascular dysfunction? And can we identify circulating biomarkers of microvascular dysfunction? Diastolic dysfunction is difficult to detect initially. And we think that the microvascular dysfunction actually precedes this, and there's no real good way of identifying this currently. And these questions are very interrelated, where answering one of these will really inform an answer to the other question. So we have been using a type 2 diabetic model, the DB mouse, which is a leptin receptor mutant mouse that are obese and hyperglycemic as early as 5 weeks of age. And by 14 weeks of age, they develop diastolic dysfunction, as you can see here from strain analysis from echocardiography. When we looked at the microvasculature in the heart, and this was a collaboration with Paul Delgado's lab at Sick Kids, at 4 weeks of age, the microvasculature looks completely normal. But by 8 weeks of age, you already have microvascular rarefaction. There's less vessel area, decreased vessel length, and an increase in mean lacunarity, which is just a measure of the space between the vessels. And this continues out to 14 weeks. But remember that the diastolic dysfunction doesn't happen until 14 weeks. So this microvascular dysfunction that we can identify is an early marker in terms of the pathogenesis of diastolic dysfunction. Now, SGLT2 inhibitors have become a very hot topic in this field because they've been shown to reduce hospitalization and cardiac death in patients with HFpEF. And so we have been using these in our mouse models, and we start treatment with empagliflozin at 5 weeks of age when we first start to see hyperglycemia in this model. And as you can see, it controls hyperglycemia quite well. In preliminary data, we find that it appears that the SGLT2 inhibitors can improve vascularization in the heart. And now what we're trying to do is really to understand molecularly what happens in the cardiac microvascular endothelium in diabetes, and how do SGLT2 inhibitors improve gene regulation. So we've been isolating endothelial cells from the heart and performing RNA sequencing to look at gene expression changes, TAC sequencing to look at open regions of chromatin, which gives us insight into transcriptional pathways that are altered, and then trying to understand really what is happening at the molecular level. Now, what we find is that looking at a PCA plot, the diabetic endothelium is quite distinct in terms of the genes that are expressed there compared to heterozygous mice, which are non-diabetic, or diabetic mice that have been treated with EMPA. There's a lot of gene expression changes that occur, but I really want to highlight changes in angiogenic markers. You can see there a whole panel of angiogenic genes that are decreased in endothelial cells from a diabetic mouse heart, and those are largely rescued in mice that are treated with EMPA. Now, if we look at the attack sequencing data, we can perform something called transcription factor footprinting analysis. And this is quite a complicated plot here, but I really want you to focus on the right-hand side, really highlighting what's changed. So we can look at these footprints, and it tells us which transcription factors are increasing in occupancy in endothelial cells with EMPA treatment versus those that are decreased. And if we look at those that are increased in binding, we find transcription factors like AP1, the ETS family, MEF2 family transcription factors, and those are very much positively regulating angiogenesis. On the other hand, there's a decrease in footprinting of transcription factors like hypoxia-inducible factor and factors that are known to regulate endothelial to mesenchymal transition like TWIST, SNAIL, and ZEB. And both hypoxia and endo-MT are thought to play a role potentially in the changes in the endothelium in diabetes. Now, this is an ongoing project, and in future directions, what we'd love to be able to do is to deliver messenger RNAs encoding proangiogenic factors to the endothelium using endothelial cell directed nanoparticles, and this is an ongoing collaboration with Dr. Omar Khan. Now, I want to shift gears a little bit from trying to understand the molecular basis of what's happening in the endothelium to think about how we might be able to find biomarkers. Now, one of the areas that we've been focused on are extracellular vesicles, and the endothelium makes these extracellular vesicles constitutively, but in response to cardiometabolic insults like hyperglycemia, they can produce more vesicles, and the content of those vesicles change. And so these vesicles can be used as a circulating biomarker of endothelial cell health. They're also very much involved in the biology, so they're able to communicate with other cells by transferring their contents. But I really want to focus on this aspect of their biomarker capability. So when we've looked in our models, there's an increase in the number and the size of vesicles, and that's quantified here. And if we look at the contents, the microRNA contents, we find a number of microRNAs that are either increased or decreased in these vesicles. And in collaboration with Kim Connolly's lab at St. Mike's, we looked at a lean rat model of diabetic diastolic dysfunction and found a number of similar microRNAs. We focused on the miR30 family, miR30D and E, because they're induced very early. So they're induced as early as 8 weeks, which is when we start to see microvascular dysfunction, and they continue out to 14 weeks. We looked at a variety of different organs in these mice, and it was really the heart where miR30 was being induced. So this may be the source of this circulating microRNA. And when we pull out endothelial cells from the heart, we find that the miR30 family is induced specifically in the endothelium, but not in the non-endothelial cells. And if we look then at pathways that are predicted to be targeted by this microRNA family, we found, interestingly, fatty acid biosynthesis and metabolism were overrepresented. There's a number of target genes that we found to be regulated by miR30. Now, fatty acid metabolism is quite interesting when you think about heart failure, because the cardiac myocytes use primarily fatty acids as a fuel source, and this has been involved in diabetic cardiomyopathy. And the endothelium is actually a gatekeeper. It controls the flux of these fatty acids from the blood into the underlying cardiomyocytes. And endothelial cells can take these up, they can temporarily store them in lipid droplets, and then transfer them to myocytes. Now, endothelial cells, on the other hand, primarily are glycolytic, but they can actually use fatty acids as a fuel. And we wondered whether this microRNA might be altering the way that the endothelial cells were utilizing fatty acids. When we performed a seahorse assay to look at how endothelial cells utilize fatty acids, we found that when you overexpress miR30, you get more utilization of fatty acids as a fuel source, and that's quantified here. Knocking down the miR30 family, we saw a decrease in utilization of fatty acids, and that again is quantified here. Now, one of the aspects of fatty acid metabolism in endothelium that's important in this context is that it can actually drive oxidative stress. So this is measuring cell rocks, and in endothelial cells overexpressing miR30, if you give them palmitate as a fuel source, it induces more reactive oxygen species. And there's also more 4-HNE, which is a lipid peroxidation marker that's produced in highly oxidative environments. Now, I mentioned at the beginning that nitric oxide was quite important. And if you take endothelial cells and you give them palmitate, it decreases eNOS. So does overexpression of miR30. And if you combine those, you get an even greater decrease in eNOS, mRNA, and protein. And in the hearts, we find a decrease in eNOS at later stages when we see diastolic dysfunction. Now, the final thing that we wanted to look at is whether if we inhibited miR30 in vivo, whether we could rescue some of the microvascular defects we see. So we injected control or miR30 LNA inhibitors at 5, 6, and 7 weeks of age, and then looked at 8 weeks of age. And although the microvascular density was only marginally changed, we did find a very significant decrease in both markers of DNA damage and oxidative stress within the microvasculature, suggesting that miR30 plays potentially an important role in microvascular health. So just to summarize the work that I presented here, and also didn't have time to go through all of this, but we do find that we think senescence drives the induction of miR30. It downregulates fatty acid biosynthesis in the endothelium, but this actually increases the utilization of exogenous fatty acids, driving oxidative stress and DNA damage that we think contributes to microvascular rarefaction and diastolic dysfunction. As I showed you at the beginning of the talk, this is coupled with a decrease in angiogenic signaling within the endothelium, and we think this then creates a situation where repair is impeded. So with that, I'll thank the members, current and former members of the lab that have done this work. This wouldn't be possible without a number of collaborations, both at UHN, Sick Kids, and St. Mike's, and also thank you to the funders. Thanks.
Thank you so much, Jason.